So basics of bioinformatics. Today we are going to see this bioinformatics overview. I will give you what this bioinformatics deals and how to make this easy to understand for your question. And this is the subtitles which I am going to cover in this paper. There is a origin of bioinformatics, introduction to basic tools, etc. So this is very important for our modern biology research bank. So if you want to understand better, you have to go for these milestones of biology which are taking place in the system. The first one is the PCR, is the polymerase chain reaction technique. So which goes like this. 30 cycles of PCR will make 6 to 7 hours and make more than 1 million copies of gene. And fluorescent automated DNA sequencing where you are going to make your 4 nucleotides to label with the fluorescent dyes of different nature, and that will be read by this laser directly to get what is that nucleotide in the sequence of the gene. So this is a very rapid and automated where your 4 nucleotides will be read, the color will be read with the laser very rapidly, just as they appear on the screen. So this results in a rapid generation of huge biological data in the form of gene sequences, and that made origination of bioinformatics. So if you look at this, what is the real origination of bioinformatics? So if you look at this slide, what you are looking is nothing but a DNA which is precipitated in the ethanol, and this DNA looks like similar in all organisms, and this will make a complete organism. So all the information to make a complete organism is available in the DNA. If you take animals, if you take plants, bacteria, or including human beings yourself also, so all the information to make an organism is available in the DNA. So DNA encodes the information. How do this information? It is present in the coded form. So this encodes information. So then, depending upon the information that is present in encoded form, it will make the plants, it will make the human beings, and it will make the rat or mice or whatever it may be. So this encoded information is referred as a genetic information, generally very broadly, or it is linked with the biology or life of the system, so we refer this as a biological information. So biological information is organized in the system in form of DNA, and then which in turn will forms the corresponding mRNAs to translation, and transcription will make them to go for this protein and do the functional activities day to day. So if you study the DNA of few groups of DNA, then it is generally genetic studies. If you study the mRNA, group of mRNA, transcription studies. If you study the protein studies. But the technology of advancement made the people to study that complete gene sequences available in the system. That is a total genes available. It may be 5,000, 10,000, 25,000, whatever it may be. So all those things are there, so which has taken to this, and then if you study them simultaneously, then a new technical term comes into this that is called as a genome or genomics. So genomes refer to this the total genes available in the system. If you study simultaneously, then it is called as a genome or genomics. Transcriptome or transcriptomics is another one where the total mRNA expressed by a cell in the given system time, it is called as a protein. And proteomics is very important. That is where the total protein is expressed by a cell in the diseased condition or in the normal condition or in the cancerous cell or while treatment of disease, so the drug action, modification of proteins. All this expression of proteins will be studied excellent in this proteomic studies. So the general capability of studies is given by this genome and readiness of the cell.